Welcome back to Concise Microbiology, an exam preparatory video with me, Kavya Tendral. Today, we'll be seeing about the biological classes of antigens and toxins. First, we'll be seeing about the difference between exoendotoxin, and then we'll go for biological classes of antigens. First, in this picture, we are seeing the pictorial representation of endotoxin. To make it more easier and to remember and correlate to day-to-day activities what we have seen, we can remember this picture, that is of human bomb. In human bomb, here he is tying the explosive around his body. Remember this. Now coming to exotoxin, here exotoxin is similarly correlated with some other picture, that is a person who is going to throw the explosive over an area. Now compare these two images. Let us now see the exoendotoxins. The basic properties of exoendotoxins are like exactly opposite. So let us remember at least one, and the opposite of this only is the other. We'll study only endotoxin. As you have studied endotoxin, the example is a human bomb. What happens in human bomb? A human will be tying the explosive over that, and he will be destroying the place after he is dying with that explosive, right? So here also the bacteria will die. There is lysis of bacteria, and there will be release of toxins. The toxins released are heat stable, and the toxins are like large doses are needed to lethal effect. Like in human bomb, when the person is explosively dying, the area which is surrounding that, like non-specifically, it will be destroyed. And here also in endotoxin, there is non-specific destruction, that non-specific affection of this toxin, and as well there is no tissue affinity. Non-specific action also, yes. So because of this non-specific action, the antibody is also ineffective in neutralization of the antigen. Okay, now exactly what we have seen, the opposite is for the exotoxin. One other important thing is usually gram-negative bacteria are the one which is going to produce endotoxin, because the endotoxin is itself the lipopolysaccharide. Now coming to that tabulation, one thing we have not discussed is the mode of action. Here in endotoxin, the mode of action is it is going to increase the interleukin 1 and TNF, tumor necrosis factor, thereby it causes febrile illness. Now coming to exotoxin, it is like mostly enzyme like action. Next, another important thing is use of vaccine. Like endotoxin, there is no effective vaccine being available, whereas exotoxin, there is a vaccine available, that is in form of toxoid. To remember, exo as X and toxoid also has X in that, so remember like that. The rest of the things we have discussed. Like exotoxin is a protein, we know. So because of this being a protein, we know protein can be destroyed or destructed. Proteolysis can be done at heat, more temperature. So there will be heat labile nature of this exotoxin. Now we'll move on to the biological classes of antigens. These are basically three types: T cell dependent, T cell independent, and superantigen. Very simple. If an antigen is processed by the APC, presented to the T cell will produce cytokines that will stimulate the mature B cell. The mature B cell will produce monoclonal it will be activated monoclonally, and there will be specific antibody produced. There is a complete immune response, right? So here there will be memory will be present. As well, there will be affinity maturation and class switchover seen in this T cell dependent. As T cell independent, here T cell is independent, thereby the APC and as well T cell is not having any role in the immune response. So thereby the antigen directly goes stimulate the immunoglobulin receptor of the B cell. Here one thing to notice is both mature and immature B cell. After being stimulated by that or activated by that antigen, there is polyclonal stimulation of this mature and immature B cell. As a result of that, there will be increased gamma globulin. There is gamma globulin, hypergamma-globulinemia, basically a non-specific antibody of IgM and IgG3 types. The same is what happening in superantigen ultimately, but what is initially happening is whenever there is a superantigen being invading the host, at that time the superantigen is going to act on, that is non-specifically bind on the MHC2 receptor on the APC and the variable chain beta receptor on the T cell receptor. So because of that, there will be a massive non-specific cytokine release In turn, there will be same, that is polyclonal B cell stimulation, hypergammaglobulinemia, and IgM IgG. One important thing, there is no memory because there is no T cell involved. And another important thing, there is no affinity maturation and class switchover. This is what we have discussed in previous, that is here it is tabulated. One other important thing is T cell independent, it is independent of T cell, so it will be slowly metabolized. T cell dependent, there will be rapid metabolization of the antigens. Coming to T cell, what are the T cell independent antigens? Are three things: capsule, flagella, and LPS. To remember it, we'll remember like CFL bulb, right? So CFL bulb, you know, it is like for saving the energy. Likewise, T cell independent is saving the action or involvement of T cell. Like that we'll remember. So CFL, C for capsule, F for flagella, and L for lipopolysaccharide. Now, how to remember the super antigens? Very simple. Just take only that super. As far as bacteria is concerned, it is this way: Staphylococcus aureus, S and U. So the Staphylococcus toxins 1. Next is Streptococcus pyrogenic toxin, S and P. Done. Another extra thing what you remember is Mycoplasma arthritis, MAM. Next is Yersinia. Here Yersinia is not, the organism here is not causing the plague. Remember this. Yersinia enterocolitica and Yersinia pseudotuberculosis are the organism which is going to have, it is acting as a super antigens. Now coming to next is viral super antigen. Very simple to remember here, because we have seen the super antigens are third different class of the biological class, right? To remember virus, if I ask you to name some viruses which has the abbreviation in three letters, and that could be EBV, CMV, HIV, and extra we need to remember is rabies nucleocapsid. That's it. So this is the third group, you know, viruses. It is EBV, CMV, and HIV. Especially in HIV, NEF and coded super antigen. That's all. These are all the viral examples. Next, coming to fungal example, here we'll take super. We add another word to complete. It is superficial mycosis, that is caused by Malassezia furfur, and therefore Malassezia furfur is one of the organisms that is having the super antigen. Thank you so much for your patient listening.